Well, let's not keep Dr. Rember from sharing what he has to talk to us about tonight. So, Mackenzie, do you want to introduce him with all of his credentials, or should we just let him?
Sure. So,
You don't need to judge. My credentials aren't that important.
at least just the basics. So tonight we have the pleasure of listening to Dr. Bill Rember. He is a professor at the University of Idaho in Moscow, Idaho. He works with Miocene plants, primarily from the Clarkia flora, which is about 15 million years ago. And I'll let him take the floor.
Well, thank you. I gave a talk in person to your group many years ago over in that little town where your museum is, that's west of Portland. What's the name of that?
Hillsboro.
What?
Hillsboro.
Hillsboro, that's right. Is that museum still your focal point?
Well, at the moment, no, because we're all meeting online for the last
of course.
year and a half. And we haven't decided if we're going to go back there or not when we finally start meeting face to face.
Anyway, it's a pleasure to talk with all you people, and I'll go ahead and start it's a slideshow, and it's made from slides that go back 40 years, and some of them are brand new. So it's kind of a hodgepodge. And like I said, I'm 79 years old, and this infernal machine is something I have to put up with, but it's nice in a lot of ways. So let me see if I can share the screen with this, and we'll see if we can get this thing started. Can everybody see that all right?
Yes, sir.
Well, I've been studying this since 1974, and I fell in love with it and have been able to acquire one of the sites, and I now have a house at 15 feet in front of my research area. So this is what Pacific Northwest looks like now, and it isn't a lot different than it looked like in 16 million years ago when before the Columbia River basalt came out. The Coast Range wasn't here then, it was in the building stage. So this was kind of in doubt from the edge of the old Cascades here. But I think the Cascades were just as high then as they are now. In fact, we have a lot of volcanic ash in the fossil site that is probably derived from the old Cascades, and they were composite volcanoes that don't really last very long in geologic history. About 16 million years ago, 16.6, the whole Western United States was interrupted probably by a shift in the Pacific Plate. If we go to the island of Midway, you'll see that there's a direct shift in the Emperor seamounts that come off of Alaska's Aleutian chain, come down to Midway and change direction almost at a right angle and come out and end up at the Hawaiian Islands now. And I think that put a lot of stress on the North American plate and resulted in big fissures opening up. And they opened up in this area called the Chief Joseph Dyke Swarm. And we used to think the bottom of the Columbia River basalt was the Imnaha basalt. But we now have added in the Steens basalt, which is another 6,000 feet or 3,000 feet of basalt, making the whole stack about 6,000 feet thick. And this is an old stratigraphy. Ignore these dates because at the end of my talk, I'll show you the new dates that we have established. The Clarkia fossil site sits right here, and it's up a valley that was dammed to the north by the Priest Rapids flow of the Wanapum basalt. And the rest of these are sites that are equivalent in age except Whitebird. At the time I made this slide, Whitebird was considered to be behind Priest Rapids basalt too, but it isn't anymore. It's a much younger site. We don't know how much younger because the dating has just gone through the end of the Wanapum at this time. Vantage, Spokane, there's one outside of Pullman, and we uncovered some doing an excavation of polluted ground near the university. And of course, Whitebird and Oviatt Creek is also behind the Priest Rapids basalt. By the way, I'm a geologist paleobotanist, but I was a forestry student to start with, and I developed a great love for trees during that period of time. I had a dendrology teacher that could pass his own enthusiasm on to his students, a very rare talent. And he instilled a love of trees with me. And most fossil plants in the Miocene are tree species. So I sort of knew them before I started working on this site, which really helped to know the vocabulary. This we used to think was a flow. It is a flow in this area, but in this area, it was a sill. It was basalt flow came up underneath the basalt, the lake, while the lake was in place. And we've drilled it in several places, and the beds are burned above and below, indicating it was a sill, not a flow. And we have a drill hole at P37. This is the site I own. It's sitting right here on what we consider an island in the lake. And I'll go on and get you to the plants. The whole story of Clarkia started one day in 1972 when a man named Francis Kienbaum was plowing the end off of that little knoll there in order to make a snowmobile racetrack for his brother, who was a snowmobile enthusiast and had a club in Spokane. And when they got through making the racetrack, he actually worked far into the night. And the next day he came out and found all these blocks with leaves all over them. The leaves didn't excite him at all, but he found this a little fish like this one. It's a fish in the minnow family, like the pike minnow of today. And he called the paleontologist at the University of Idaho, and he indicated that he'd like to come out and look at it because of the leaves. He was a paleobotanist, Jack Smiley. And this is the sort of leaves that he would find in an average slab. This is hard to believe, but the average slab at that site will look like this. And this is a tulip tree leaf. This is an oak leaf. This is a chestnut. This is a tupelo leaf. This is sweet gum. That's bald cypress. And another sweet gum up here. And an alder right here. It's a fantastic site. It's almost like they're laid in there so we can see each one of them. There'll be 50 or so leaves on each flat surface. This is what the site looked when he finished the racetrack. It's now been converted to a motocross racetrack, and he actually has 2 national meets a year here with 4-cycle motocross vehicles. The site, the preservation of the oxidized material is really phenomenal. You can actually see the ultimate venation in most of the leaves, even in the oxidized one. But the oxidized ones are beautiful. They're as pretty a fossil as you'll ever see. This is a platanus leaf, but this drab thing to the right here is a compression of a leaf. of platanus. And we can take that leaf and look at the chemistry of it, and it's very likely to be like the chemistry of the modern genus Platanus. This is a sycamore leaf. And I've developed a method to lift this leaf off of the rock and study it as a biological specimen rather than an imprint like this is, which gives us a lot more information. I had a lady that I helped with a study she was from Stockholm, Sweden. They're studying the mid-Miocene climatic optimum in order to look at a climate that we may be heading to with global warming. I'm going to start at the bottom of things. We used to consider this a plant. Actually, when I started school in 1960, it was a plant. They're fungus now, and we now know they're closer to related to you and I as they are more than they are plants. These are the hyphae, the pycnidium for the spores are in here. This is another fungus. This is a cleared bean pod that I found a lot of fungi on, and I photographed it for the fungi. This is an epiphyllous fungi that grows only in warm, temperate, subtropical and tropical areas today. So it's a good indicator of the temperature of the climate at Miocene Clarkia. These are lower plants. That's Isoetes quillwort. These are Polypodium ferns, and this is an Osmunda fern. And these are bryophytes, mosses that grow on. This is a piece of bark. The bark fell off and brought the bryophytes into the lake and were preserved as fossils. This is a very interesting fern, and I could see when I found it that it was a fertile fern, and you can actually see these sori right here. They're curled under the leaf, kind of like the maidenhair fern is. And I lifted it, and this is what it looked like when I brought it off of the rock. And this shows you the exquisite preservation that we have at this site. And I've been working with Steve Manchester at the University of Florida, and he took one of these sori right here and put it under an SEM and sent me the pictures. Now this is the sori, one of those sori, and these are sporophylls, which are full of spores. And you can see the sporophyll close up here. Now, this is a close up of the spores inside the sporophyll. And here is a close up. These are very silate spores, very smooth with a trilate spore. And we'd found these a long time. Now we know what the fern was that produced them. This is very rare to be able to find the spore that goes with the plant. These are members of the Taxaceae, which is a yew family called Amenotaxus. These are much longer than the yew tree of the Northwest today. And they live only in China today. This is a cleared leaf of one of those yews, and you can see the number of stomata that are in it. So, and again, keeping up with the climate being different, there are about half the number of stomatal bands in the fossil leaf as there are in modern ones. These are members of the old bald cypress family. They've been combined in with the Cupressaceae, which is the cypress family. And these are cones and foliage of Cunninghamia. This is a cone. This is Metasequoia, one of my favorite trees, and a cone. This is Glyptostrobus, the Chinese fir, and the Glyptostrobus cone. And this is a foliage that we used to call sequoia, but I don't think it was sequoia because we've never found the cones of sequoia, and we found the cones and foliage of all the rest of these. I think this is just a variant of taxodium, and this is a cone of taxodium that dropped off before it fell apart. This is a cleared leaf and a micro photograph of the Cunninghamia leaf. Again, this is an Asian species and this is exquisite. This looks better than the actual living leaf with the stomatal apparatus. And again, there are half the many of stomates on this as there are on the modern one. This is Metasequoia. This is the Cunninghamia I showed you. And they fall off in branches. So we find these branches. If you go to the University of Washington Arboretum, you'll find a little grove of these. And underneath the grove are the branches of these. They don't fall off in needles like most of the trees do. They lose their branches. This is a hodgepodge of conifers. This is an incense cedar like Northern California. This is the cone of it. And this is more like the one that lives in Asia. This is Amenotaxus again. This is Taxus, the yew. This is like the Port Orford cedar in Oregon, Chamaecyparis. And this is a foliage of what we used to think was Abies, which is the fir. But we now think it's Cathea, which is rather like the Metasequoia. It was found growing in a valley in China. It had been discovered in the fossil record before in Hungary. This is what I thought used to go with this thing, but it doesn't. We now have the modern one to look at, and I'm working with a grad student now taking my pollen course, and we're going to do an experiment by looking at the pollen in these and associating the pollen with the catkin or the female or the male cone that comes with the conifers. These are all the members of the pine family. There's two different subgenera in the pines. The Haploxylon or white pines, or Diploxylon or the red pines. A short turn-needle pine, a three-needle pine, and cones and male cones. This is a cone of a white pine. You can see the little yellow part. That's resin. That's a scale of a white pine, much like the limber or the sugar pine in Northern California and Southern Oregon. This was a cone that I found and dissolved out of the rock with HF, and it is almost a dead ringer for the Apache pine or the Mexican white pine found in New Mexico and Arizona, as well as the country of Mexico. This is the backside of that same cone, and these are patches of resin now. And I'm working with a biochemist, a geologist biochemist that works on organic material, and she's going to be working on all of the organics from now on, so we can rate the maybe get a really good identification of these through the resin that's produced. This is the modern one. There's only 4 cones of all the pines in the world that have reflex cones on it. And this is almost a dead ringer for that cone I found in the fossils. This is a cone that's very similar to the ponderosa pine of today. Now we're in the diacots. This is an Asian species called a wingnut, Pterocaria, meaning wingnut. And you can see it is in the Juglandaceae family. It looks just like a walnut. This is another wingnut called Oromonia, found in Central America today and in Asia. The only evidence of hickory we have, and we have lots of evidence in the pollen record, this is an SEM picture of a Carya pollen grain, very distinctive of the genus Carya. There's no doubting that the Carya was here, but I've never found it in the fossil record. This is the magnolias. I've got one like the cucumber tree, a northern magnolia, and then this is very much like the Virginiana, the Magnolia virginiana, which is the southern magnolia. This is a fruiting body, a female fruiting body of magnolia that's very much like the modern one. You can see the modern one for comparison. Another member of the magnolia family is the tulip tree, Liriodendron. And we have the cones, not cones, female fruiting bodies. My botanist friends chastised me for calling these cones, but they look like cones. This is a modern one torn apart to resemble this one. And then we find the individual seeds like this one. Very much variation in the leaves, and we have all the variations in the fossil that we find on modern leaves today. These are members of the Lauraceae, the laurel family. There are two main members, and rarely we find sassafras. This is the spice bush or lindra, and this is Persea or the avocado. The Persea americana is the modern avocado, and it looks identical to this leaf. However, there's a lot of members of the Lauraceae in Asia, and quite frankly, it could be a number of, because the leaves are nearly identical within the family. These are vines that grew in the forest of Lake Clarkia. This is a grape. This is no longer a vine, it's actually a tree. When this slide was made, we thought it was a Carolina mood seed, but Steve Manchester has found the pollen on the fruits that goes with this thing, and it's an extinct genus in the Trochodendraceae, a very ancient bunch of angiosperms. This is a rose leaf, and this, some of you in the audience are old enough to remember the Song of the South. Uncle Remus and Brer Rabbit, Brer Fox. This is the briar patch of Brer. This is the leaf that goes with the briar. This is the briar vine that grows in those briar patches in Kentucky and Tennessee. I wish that movie was still available for our children, because growing up in Idaho, I'd never seen a black person in my life. And Uncle Remus in that movie was a very positive depiction of a black person to me. And the tar baby had nothing to do. I didn't even register to me that it might be a black person. And that's why it's socially unacceptable to show that movie anymore. One of my pet peeves. This is a cleared leaf of Smilax. And this is an interesting flower. We still don't know where its affinity is, but Robert Rose, I guess, is part of your group. He found cross sections of this in his site up about outside of Sweet Home. And Steve Manchester had a flower from Japan, and he had some from Sucker Creek in Idaho. And he was writing a paper on this flower when I found it. And the night I found it, I took a picture of it and sent it to Steve. And he had all the reviews back on his paper. And 30 days after this flower was found, it was published in a professional paper. And that's about as fast as you can get it done. This is a monocot called Caldesia. It's an aquatic plant that grows up to Estonia in Europe and down into Africa. Goes from almost boreal forest habitats all the way down into tropical habitats in Africa. It's an Asian, and there is quite a few in Eastern Asia as well as Western Asia. This is a member of the Platanaceae, the sycamore, and this is sweet gum. And this is one of my favorites, as you'll see with the next slide. This is the one we use for the logo of the Miocene, or the Tertiary Research Institute here at the University of Idaho. This is the fruiting body here, and this is a replacement cell for cell with limonite of the organic material. And if we were looking at the abaxial part of this leaf, you could see the stomata apparatus in it. It's that well replaced. This is a cleared leaf of Cercidiphyllum crenatum, which is the fossil member of the family. It's one of those old angiosperms that goes all the way back into the Mesozoic, the late Cretaceous. These are members of the birch family. This is Betula. This is Austria. This is an Austria fruit. This is actually an alder that looks a lot like the alders you find in Japan and in Mexico. And they're evergreen alders. And these are the female fruiting bodies of alder. These are the two different types of birch we have here. This is Betula lenta, much like the sweet birch or Betula alleganiensis that grows in the Appalachians today. And this is Betula ferrat, which is very much like the Asian species called Betula maui-moixii, which is Maui-Moix was a Polish botanist that first went into Europe in the late 1800s and has a lot of species named after him. This is another alder that we haven't figured out its affinity yet, but it's a beautiful specimen. And I just have one talk. These are members of the Fagaceae, Castanea the chestnut, Fagus the beech. This is a beech fruit, and this is an extinct genus called Pseudophagus that my major professor, Jack Smiley, described in 1976 with Maurice Huggins. And they did we found probably 50 or 60 leaves with this fruit laying across the leaf. So we know the fruit was attached. It's attached right at the axial of the leaf, much like this fruit is attached. This is two fruits in a involucre. This is there's a little involucre around the top of this that holds one nut that is very similar to this. So we think it's certainly in the same family, but there's no Fagaceae alive today. And we have oaks. This is much like the tan oak that grows in Southern Oregon and all through California. This is black oak, the only oak we have. We have no white oaks like the Oregon oak or the live oaks from the California. And we do have acorns. This is the Salicaceae, the willow family, which includes poplars. We have about 3 or 4 different types of poplars. This is much like the eastern cottonwood. This is somewhat like the big tooth aspen in the north part of eastern United States, related to our quaking aspen. These are willows. Willows are quite rare, and I can't tell you why that is. We found quite a few species of it. A friend of mine, Walter Buchler, has written a paper on fossil willows that I don't know where he got it published. I'm pretty sure it's published, but it's I was able to clear a leaf for him and send it to him for that paper. This is another hodgepodge. That's probably the only leaf of basswood or tilia that we have. This is a partial leaf, the only one we had when I made these slides, of Aeschylus, the horse chestnut. This is the only leaf I know of this one is the only leaf I know of Zelkova, and this is an elm. We have elm pollen, but very few leaves. Probably like the hickories, they didn't live in the same area as the pollen had spread to. And this is a hydrangea with a hydrangea blossom, and I'll have a closer shot of the hydrangea blossom. This is this is the sterile blossom that comes on the side of the snowball bush. You see around a lot of professional buildings, especially in the Northwest, because hydrangeas work very well. Now, some of them have the blue flowers, and this one would have been a beautiful horticultural variety, because this is a meter scale along the side. So this thing was one of the blossoms, which are about a little bit bigger than the size of my thumbnail now. This one was nearly 2 inches long. These are pods and leaves of legumes. We know legumes are there because they have an interesting thing called a pulvinus on their base. And it's a wrinkle that allows the plant to pump water in and out and change the angle of the leaf, either to hit more sunshine or away from the sun if it's getting too much sun. And these small leaves go with these big pods, which is the swamp honey locust. And you can't, oops, I'm sorry about that. Oh, I see. That pod at the top, I'll put your pictures down a little bit lower here. This is a pod that goes with these bigger leaves. And we don't know the pod. We have some odd ones, but we don't have enough to make a stratigraphic association with the pods and the leaves like we do these pods, these leaves, and these leaves with this pod. This is a member of the cornales, the dogwood family, or the dogwood order, and these are members of the Nyssa, the swamp tupelo, and this is the mountain tupelo type. This is a fruit of the swamp tupelo, and actually if this photograph is a little better, you'd see the ribbed seed inside an involucre of fruit. The fruit is a berry, and that fruit is still preserved in the record. This is a close-up of that swamp tupelo leaf, and along with a leaf from that area of my fossils that has the replacement of the thing. You can see a lot of insect mining in this oak leaf that I removed carefully to expose the rest of the tupelo leaf. Again, you can see a little insect mining. Almost every leaf has insect mining on it. And I have a graduate student that if he finishes his master's, he's going to do a PhD with on the insects there. He's studying for a master's down at Arizona State now. The maples, we have several different maples, several different samaras, and associating the maples, I'll talk a little bit later about that when I get into the strike. This is a fossil samara that you that live in the Northwest are probably familiar with. It's almost a dead ringer for the macrophyllum, the big leaf Samara of Acer macrophyllum. And it's a very common tree all the way down through Oregon, Northern California, and up through Washington. And actually it was called, Jack Wolf did a paper on the maples of the Kenai Peninsula in Alaska and called this Acer macrophyllum. Later on, he sort of backed off of that. So I don't think we can, with 16 million years of evolution, I think there must be some genetic change in those fossils in order that the same species wouldn't be around today. We have no way of looking at that until we find genetic material from the Clarkia fossils, which people are working on as I speak. We have insects there. This is a flying ant. This is a beetle. That color is not pigment. It's 3 layers of cutin that has strips in it, much like a CD does. When you hold a CD up to sunlight, it breaks the black or breaks the colors down. And there's a lady named Maria McNamara who came to my place to find beetles. And she wrote a paper in the London Philosophical Society Journal on the color of fossil beetles. And it's an outstanding, and she used this fossil in that paper. I donated it to her when she came to visit. We found 12 beetles at my place the day she came, and beetles are rare as can be in the fossil record. So it's just a wonderful Lagerstätten to work in. This is the only arachnid that's ever been found there. It's much like a trapdoor spider today. And this is a fish that was described as the genus Huco. And for you fishermen that may know of the taimen it's in the rivers that grow into the Arctic Ocean. It's an anadromous fish that lives in the rivers, both the rivers that go into the Arctic Ocean as well as the ones that go into the North Pacific along with the salmon. It's called Huco, and it was written up by Jerry Smith, a paleoichthyologist. This is what the track looked like when I did my dissertation there. In fact, these are the blocks that I cut out with the Pulaski, starting over here and going through about 9.5 meters of section. And I went through it a layer by layer and counted over 11,000 fossils. And this is the section I went through. The fossils end right at this this level right here. So I went through and I started it. I had taken a course before I had any stratigraphy. I took a course in palynology at WSU, and they always start at the top with 0. Geologists always start at the bottom at the base of the site. I didn't have the base of the site here, so I started. It's caused untold amounts of problems with fossil people because stratigraphy always goes the other way around. And I'm in the process working with a stratigrapher from Eastern Washington to turn this around and convert everything to the correct way up now that we found the bottom of the site through drill holes and excavations. I'm going to go through some of this. This is the turbidite that came in, and we have 2 major finds in the turbidite, which one was Aminotaxis and the other one was Cunninghamia. And these are from my site at Emerald Creek, because the only ones we found were the ones at the racetrack were just in this turbidite, and they weren't photogenic like the ones at the Emerald Creek site. Chamaecyparis, I'll go through these really fast, are both found at the bottom and the top. This will mean something later. Section B is a section that's about 100, 150 meters to the east of section A, and I duplicated the top to see if it matched the section A, to see that I could look at leaves and see that they were homogeneous over a length of area. This is the Port Orford cedar. These are Taxodium the most. Also, these are the individual sections that I divided it up into 30 centimeters sections and went through 5.78 meters of section, or no, 7.58 meters, excuse me, all the heck. I'm getting a little tired here, folks. Anyway, if somewhere along here they found they occurred at 5%, which is double what Dr. Ralph Cheney did in the Briggs Street flora in Central Oregon. He thought if it occurred at 2.5%, it was a dominant one. If I'd have used 2.5%, I wouldn't have had enough room for all of the major players. So I doubled that in order to cut down on the number of major players I had to deal with. This is the organs of the pollen organs. Again, it's the most common thing we found and is very dominant in the top and in the bottom. Metasequoia is dominant pretty well through. It doesn't make much difference where you check it. Pinus is the same way. It's dominant right after that, and then it tapers off fast, which is same as ABs. ABs we now know as Cathea. I look at these as, because these ash layers came in late, this one had a lot of available land close to where the organs were being lost. So those organs were on a new piece of land, and they were probably pioneer species that were crowded out later by other species. This was Cocculus. We now know it was a tree. This is the one that Steve converted to. He called these Nordenskeolea, and this is Zeuxipoides. I have to find these attached to these leaves before he'll put them together. But stratigraphically, they occur together. Magnolia, we've talked about that before. It's just like the others that throughout, as is the Lauraceae. And liquid ambers the same way. This is unique. Platanus is only in the bottom, and we'll talk about that later. We have Betula only in the bottom and only in the top. Pseudophagus is pretty much right here in the middle. That makes something. Castani is the same way. Lithocarpus is down here in the bottom also. Quercus is dominant in the middle. Cladicea is in the bottom. Zinnia is in the bottom. Sympicosalesia is kind of in the middle. That flower goes with this leaf. Again, my friend Steve in Florida doesn't believe that these are together. We'll figure that out if we can find more evidence. Nyssa is only in the top. Acer Cheney, which I believe these samaras go with these leaves, even though Jack Wolf has this samara going with these leaves. I talked with Jack about it, and he believes that that's probably true, because I found quite a few leaves in this area with these samaras, and only one or two of these samaras to go with the leaves. This is sequoia, the one we only find above it. And again, I think it's a variety of this, if you can see it right here, is the cone of sequoia. It always comes off the tree in a branch with needles on it. And we would find these in the record because we have record of all the other cones if it were there. Now, putting all that back together, the swamp species are here where the new land was, and the swamp species took over here in the end. The slope species, oaks, chestnuts, pseudofagus was extinct, cocculus was extinct. They all occur right here in the middle, whereas the riparian species, all the species of platanus, the sycamores, are worldwide are riparian species. Chamaecyparis is a riparian species as well as a swamp species. Betula is a riparian species as well as a swamp species. Lithocarpus and these must be associated with those river bottoms also. Here you can see that those, this section B sort of occurs, that matches well enough for me to think that this is, this method of analysis works. I patterned this off of a course I took in palynology in order that I could do macrofossils the way people do pollen counts. Like there's a big group doing paleoclimate studies at the University of Oregon now. I can't remember the fellow's name. He's head of the department down there in the geography department that are doing some big studies with pollen. I did it under Peter Maringer at WSU, and he was probably one of the finest scientists that I've ever worked with as far as keeping the scientific method in sight when you're doing your work. If we look at these floras worldwide, they occur on the west side of the Sierra Nevada mountains in Central California, along the Pacific coast all the way up to the Oregon-Idaho border, Oregon-Washington border, around the old mountaintops in Mexico, not the new volcanoes. These are the cloud forests, and they have the same mesophytic flora that grows in the southwestern or eastern United States today. There's a little valley in western Turkey that's subsurface irrigated. It's a glacial valley that has the same Miocene flora in it, as well as the Caucasus Mountains. There's also a patch that goes across along the Caspian Sea and then along the Himalayas, very minor and spotty. But the major site is this site in Eastern Asia. The Metasequoia forest is right here, and it is 65% of the fossils I find are growing in and around the Metasequoia forest today, or was before the Cultural Revolution. The Cultural Revolution wiped out a lot of forests in China because they had to come up with so many pounds of iron, and they cut down trees to make charcoal in order to smelt the iron. The southern tip of Korea, the island of Formosa, and the northern half of the main island of Japan are modern equivalents of the Miocene flora. This is the temperature. That's about 65 inches of rainfall a year spread out. It's summer wet situation, and the temperature never goes under, the average temperature never goes under 0 degrees, but I'm sure there were some of the colder months it did go under, because it only averages 2 degrees, 3 degrees, 1.7 in January. So this is a little valley that they took the average temperature for 5 years in the closest town to the Metasequoia forest. This is an old chart a friend of mine did for me about the Miocene climatic optimum. And this is, these dates are not quite accurate. This has been compressed by Castbaum and Schoen, and I'll show you quick like that. But I want to show you that this climatic optimum, this has been set back just 200,000 years. If you look now at where they got dates on the Steens, Castbaum was Schoen's grad student. And in 2018, they published this elegant paper on the dates of the of all the basalts up to the top of the Priest Rapids, which is in the Wanapum. Now, Castbaum was told to quit her study, and if she gets her own lab again, somebody may do the Saddle Mountains. But they only account for about 1% of all of the volume of the thing, whereas the Grand Ronde accounts for 93% of it. So this, it comes out at about 16 million years, but that's now the top of the Wanapum. And the bottom of the Steens is 16.6 million years. So the whole Columbia River basalt has been compressed into 700,000 years if we take this 16 or 15.9 date as the average. And their dates are good. What Castbaum did is collected soil surfaces and all of the inner beds that she could find exposed in the Columbia River Plateau up through the top of the Wanapum. This is an elegant paper. This paper right here, Castbaum and Schoen, 2018. And I'd be glad to send this to you. I've got it on file, and I'll give you my email account. It's bill.rember@gmail.com. And I have a lot of these papers on file that you're welcome to have. This is an elegant paper. She theorized, or Schoen theorized, that the only way a zircon could get in these soil surfaces is through volcanic action, because they're heavy, too heavy to blow in the wind. And if you didn't sample near an old riverbed, you wouldn't get zircons from anywhere else. So she took many pounds of soil surfaces from between these layers of basalt and analyzed them for the zircons, sorted the zircons. And it's just an elegant paper. I love it. This is the PaleoMag data for the sediments at Clarkia versus the basalts in the area. And you can see that they're virtually statistically identical. And I'd like to acknowledge Jack Smiley, who I learned much paleobotany from. Ken Spinkney and Jay Bobeck both introduced me to this infernal machine we're talking through. And I appreciate it a hell of a lot, but it is very frustrating for me to this day. And thank you very much, and I'll be glad to answer any questions some of you might have. So why don't I get rid of this thing and see what we can do with just talking to me. Now, can you turn this so I can see your face up close when you talk?
Yeah, it's just, I guess, so if people have questions, now would be a good time for questions.
Could you
I
talk
got
a little bit about what clearing the leads meant?
Who am I talking with?
I'm Greg Carr.
What did you want to know about clearing them?
Yeah, what is the process?
Well, I use a very bad acid called hydrochloric acid or hydrofluoric acid. That's the acid that will eat glass, and it's been banned from most universities because it is so dangerous. However, you know, cars are dangerous and firearms are dangerous. And if you know how to use it, it is perfectly good to use in a hood. And I break the bond between the clay and the leaf with the hydrofluoric acid. It takes about 5 minutes maximum to break that. And then I just carefully detach the leaf from the rock, and the hydrofluoric acid will break the silt down into almost a molecular size. And I learned that taking the pollen course at WSU, because that's how we treated the pollen samples. We dissolved them in hydrofluoric acid for overnight. I leave my samples in that I do pollen out of the Clarkia fossils. I still teach a course in pollinology. I've taught my last course for the 5th time this year. And I use samples from the Clarkia site. And we leave them in for an entire week so that it knocks that silt down to molecular size. Then we can suspend it and pour it off. Anyway, back to the leaves. Once I get the leaf out, if it's an exceptionally dark leaf, I will use hydrogen peroxide buffered to get rid of the acid. I buffer it with sodium bicarbonate, concentrated, and that allows oxidation to take place a lot sooner than if you have it in an acid condition. And you have to watch that because some of the lighter leaves will disappear right in front of your eyes. Does that answer your question?
Yes, it does very much. Thank you.
Then I actually take it and dehydrate it into 100% alcohol and put it in a mounting media. Permount has been sort of bad because it starts to oxidize and the plates go bad, like the cleared leaf plates that Jack Wolf made while he was working for the GS are all oxidizing away as we speak. And I don't know if they've got a better medium now, but they've sort of banned any toluene or benzene ring solvents. So it's really hard to find one that hasn't been banned by the safety people at universities.
Okay.
I mount it in glass, but now I just mount it in glycerin and leaves. Actually, one of your members used to come over about every other year to my site, and I allow you to collect, and I don't charge any money to come. So you're welcome to do that if you want to revise that pilgrimage everybody made from Seattle down through Portland to my site. So
yeah, we'll probably take you up on that once this COVID
yeah, that's
goes
the
away.
problem. We're outside and I require everybody to camp and bring your own food for the night or two that you stay. It's usually on a weekend because of people at work. And again, if any of you want to come and visit, just email me. You can get my email addresses from Mackenzie.
Bill?
Any other questions?
I think Peg was about to say something.
Yeah, Bill, this is Peg Johnson. I was over there and I still have my cleared leaves.
Oh, is that the one that was on display when I was at?
Right. And they still look good today.
Let's see, where, I wish I could see your face. So is this you here?
That's me right here. I don't have my face out.
Oh, okay. Are you with your husband side by side?
No, that's Linda.
Oh, okay.
Mine is just my name.
Oh, okay. I'm sorry.
She doesn't
No,
have her
that's
camera
okay.
on.
You know, I use that leaf that I gave you between the things because I think I told you to put it in glycerin
Glycerin.
every once in a while, and it's still as good a shape as it was when I gave it to you.
Yep.
How many years ago was that?
Oh, man. I think it was the first year we went over.
Must have been around 2000.
Yeah. It was a long time ago.
Yeah. Well, that's good to know. I'm glad that you're still with us.
Yeah, barely.
That's wonderful. Actually, the guy that I just see over here, Steve Manchester, who you all may know, I don't know. He's the one that turned me on to using the overhead projection material for for the cleared leaves. So anybody that visits, I usually give a cleared leaf to because they're quite common and it's easy to lift them if I've got a small crowd. I don't have to worry about the acid. My hood is a big Doug fir stump out in my front yard, as you know. Any other questions?
This is Curtis,
Yeah.
and that was amazing. Just real quick, I had no idea you could lift that kind of resolution of fossils. I was totally blown away. Thank you very much for the presentation. Big time.
Yeah, you're quite welcome.
So, Bill, this is Aaron Courier again. One of the Metasequoia fossils that you were showing had much longer needles, if you want to call them that. And you said that was also from China. Are those living today? Are there Metasequoia?
Cunninghamia?
Well, no, I don't think it was the Cunninghamia. It was in your slide presentation, and they were much longer. It wasn't just the straight ones, but it was like all curly. Do you know which one I'm talking about? I think you referred to
Oh,
them.
yeah, are you talking about the Cathea?
I think so.
Yeah, that's the, and that's still up in the air whether it's Cathea or not. I've got to do a study on that. I went to are you from Portland?
Salem.
Salem. Well,
Yeah.
you know where the botanical gardens are at Silverton, don't you?
Yes, yeah, Oregon Garden.
Okay, they have a specimen tree of Cathea growing in that garden. And I was able to go over there with Robert Rose and collect needles and male catkins so I can get the pollen out of it, which I haven't processed because right after I did that, well, not right after it, but I haven't gotten to the project because I had a heart attack that kind of limited me for a couple of years and still has me a little bit under the weather, but I'm living on bank or house money now, and that I'm 79 instead of 74. So every day is a good one.
How do you spell Cathea? So I can
It
look
was
it up.
Cathea, and I need to do that project, but I've got many, many projects to do, and I have projects I'm working on with other people that take priority of the many bucket lists that I have that are checking off before I get done.
How do you spell that so I can look it up? How do you spell?
Yeah,
Cathea?
look up Cathea and go to Silverton and see if you can find that tree. It's really neat. Actually, the guy that turned me on to it is an Oregon State taxonomist. What's his name, Steve?
You're on mute.
He was in the van with us when we were on that field trip with you. Oh golly, I can't remember names as well as I could a while back. Anyway, he teaches, he's head of the herbarium there, and he's a He's a geneticist. That
Is it Aaron Liston?
Aaron Liston, yes, exactly. He told me about it. He showed me the little one he had growing in that courtyard that's by the biology building. And then he showed me that there was a tree, quite a big tree growing in the botanical garden at Silverton. And I was on my way up to visit a geologist for the BLM in Portland, or the Forest Service, that was trying to rewrite the right regulations for that bill that got passed that made all paleobotanists have to get permits to get fossils on federal land, which I'm still very disgusted about. But he got so disgusted about it, he retired and turned it over to a vertebrate paleontologist that has no time for paleobotanists. That's my soapbox for tonight. Any other questions, or shall we say goodnight?
All right. Well, thank you again for coming and speaking with us, Bill. We appreciate it.
Well, I'm glad I could, and I wish I were doing it in person, because it was a lot better to do it with a slideshow and PowerPoint than it was to do it otherwise.
Right. Yeah. Yeah.
Thanks, Bill. You're awesome.
Thank you. I appreciate the.
We had 25 in attendance to listen to or 26 people in attendance to watch the lecture.
I want to mention one thing, that paper that there's a paper written on that fern, and Steve is a member of that paper. The rest of the people are Pinson, Manchester, and Sessa, SESSA, and they're all members of the University of Florida. I guess you're all gone.
We're here. Nope, everybody's here listening. Steve.
Well, can you get that paper to them, Mackenzie, if you're still listening to me?
Yes. Yeah. I have a copy of.
Yeah, get it to anybody
Jerry's
that wants
paper.
it, because they named it after me, and it's kind of hard for me to talk about it. So,
All right.
Great. Thank you again so much, Bill. I really appreciate you participating, and those of us who were in
yeah,
attendance.
sorry it goes so long, but that's just.
No, it was great, and we appreciate you being here, and we appreciated having you in person. I remember being here when you were in person too. So
Yeah, well, I'll be glad to do it again if I'm still alive the next time it comes
I
around.
hope so.
And talk to the people about coming over, and tell them they're all invited to come over individually if they can work it into their schedules.
We'll try to plan a field trip for this summer.
Yeah, that's a good idea.
Sounds
Great.
good to me.